This is Bernard Paulson. I will, in this lecture, discuss how to estimate numerical values for some of the parameters that we need for building or for constructing dynamic models in biology. I will also, upfront in the lecture, talk a little bit about order of magnitude estimation procedures. We have completed part 1 of this book by going through lecture 6, and we are now on to part 2 of this book, where we move away from toy models and start looking at features of models that are biological. And this includes looking at the actual numerical values of many quantities. It involves looking at the consequences of the specific structures of the stoichiometric matrices that describe biochemical reaction networks, and it involves looking at regulatory phenomena that are so prevalent in biology. Now, on the next slide, I have a little video clip that zooms in from objects that we are familiar with, like a coffee bean or a grain of rice, into increasingly smaller and smaller objects, so you can get a feeling for the relative size. In the upper left-hand corner is a length scale, and then you can see the bar at the bottom of the image, or the images, moving across the different size scales, and you can see how big they are. And this film ends with looking at the carbon atom that we know is the key atom in building biological molecules. You may have to play this clip a few times before you get a good feeling for the relative sizes of the objects shown. An actual understanding of the length scale hierarchy in biology is important, and so is an understanding of the time scale hierarchy, the abundance hierarchy, the hierarchy of connectivities, and so forth. The size scale hierarchy is the easiest one to understand, as it relates directly to the size, the physical size of the objects involved. So play this movie a few times and try to identify the objects that it zooms by and get a feeling for their relative sizes. Now, there are processes that engineers use to get estimations of the numerical values of various quantities. Many of you have probably been confronted with questions like this one: how many jelly beans are in that jar? Well, how do you go about estimating it? It would be nice to estimate the volume of that jar, try to estimate the volume of each one of the jelly beans, try to estimate the volume fraction of the jelly beans, and then calculate. There would be an educated guess. The other way of doing it is to simply to guess. Now, in science and engineering, guessing is not so good, but educated guesses are better. And let's see how these are made. So the content of this lecture is to go through some overall observations of how we do estimations. And I should state that the ability to do these well is one of the more valuable skills that you can acquire, and it takes practice. You have to do it over and over and over again before you get good at it. kind of like playing an instrument, I guess. Then we will try to estimate a few quantities associated with metabolism, typical concentrations, typical fluxes, half-lives of molecules, and power densities. Although I think I will skip the slides on power densities in here because the lecture is already a little long. Then talk a little bit about macromolecules, the typical sizes of genomes, protein concentrations, the production rates or fluxes of macromolecules, and their turnover times. We can move on from there to estimating whole cell functions like growth and other phenotypes based on what will be shown before, and then finally go through a little bit of a summary. And I have a little appendix or an addendum to this lecture that is very important. It helps to illustrate the magnitude of the quantity $1 trillion. Okay, some of the key concepts here. So we can estimate typical or characteristic orders or magnitudes for key quantities that we need to characterize cellular functions, and we can estimate these based on data that is available to us on cell size, cell mass, cell composition, metabolic complexity, genetic makeup, and so forth. We can find such information in many databases that are now available on the web. And based on this information and the procedures we'll discuss, we can get useful estimates of fluxes, concentration ranges, kinetic parameters, power densities, and so forth. Now, order of magnitude estimates and calculations are the hallmark of good engineering because one should be able to bracket a solution ahead of time. Enrico Fermi, that I think everyone knows about, was a master at this and became famous for getting answers quickly through back-of-the-envelope calculations, and therefore such calculations in some circles have been called Fermi problems. So we can go through an introduction to what a Fermi problem is or what an order of magnitude estimation problem is. So one classic Fermi problem is, and you can find it on the web if you search, is to estimate how many piano tuners there are in Chicago. Of course, a lot of the work that he did was done in Chicago. So here's a way of doing this. So I'm going to walk through a few quantities and then a few calculations and then come up with the estimate. So first, we somehow need to figure out how many pianos there are in Chicago, how many tuners there are, how quickly a piano tuner works, and so forth. So we're going to estimate, or it was estimated at the time this was done, that there are 5 million people living in Chicago, 2 people per household, and that roughly 1 in 20 households had a piano that was tuned regularly. And regularly tuned pianos are tuned once a year. Now, it takes a piano tuner about 2 hours to tune a piano, including travel time from one job to the next. Every piano tuner would work 8 hours a day, 5 days a week, 50 weeks a year, and that's of course true for most jobs. But from these assumptions or these statements, we can now do our estimate. First, we can calculate how many piano tunings there are per year in Chicago. And so we take 5 million divided by 2, To get the number of households, a piano in every 20 households, one piano tuning per year, and we calculate the market size here is 125,000 piano tunings per year. We can then similarly calculate how many jobs the average piano tuner performs per year. So we take 50 weeks per year times 5 days a week, 8 hours a day, one piano tuning per 2 hours, and calculate that each tuner can tune 1,000 pianos per year. And if you ratio the two, you estimate that there should be about 100 to 125 or so piano tuners active in Chicago at any given point in time. So this is an illustration of how these sorts of calculations are actually done. Now, the significance of this is that we can estimate key biological quantities based on knowing a few foundational facts and simple ideas from physics and chemistry. Numbers that are collected by scientific community that seem unrelated can be brought into a coherent whole by some of these calculations. And a lot of these numbers, as I'll show you, are found in different websites that you can access. So in this lecture, I'm just going to go through a couple of examples. And these are prepared by Josh Lerman, a graduate student here at UCSD. First one is, how many proteins can be produced from a single messenger RNA in E. coli? Very important number in biology. How many protein molecules are made from a single transcript? Another one that is interesting is to estimate how many ATP syntheses are needed in a cell to produce all the ATP that is required to drive growth in a cell like E. coli. E. coli is used for most of these types of calculations for the simple reason that so much is known about it, and it's frequently used for a variety of experiments. So let's proceed first with this estimation of how many proteins are made per messenger RNA. So first, a few facts. So the average length of a gene in prokaryotes is a little over 1,000 nucleotides. And remember, there are 3 nucleotides per codon, and so you divide that number by 3 to get the average number of amino acids per protein. Ribosomes are on the order of 20 nanometers in size. So if you take these 1,100 nucleotides, you can only bind a ribosome at the best of times every 50 nucleotides. So there is really a space for only 22 ribosomes working bumper to bumper on the same transcript. Now, the rate of translation, the rate of peptide bond formation by a ribosome is something like 12 to 20 amino acids per second. We'll pick a midpoint here of 16. So 22 ribosomes working together on a transcript could be incorporating up to 350 amino acids per second. And as I said, if you take the 1,100 nucleotides and divide by 3, you get the average amino acid length of a protein to be 367. So the effective translation speed here is that you can make 1 protein per second from the transcript. The half-life of a transcript in E. coli is about 60 minutes or 360 seconds. So you ratio these two and you figure out that you can get about 500 protein molecules at the very, very best of times from a single transcript. So that becomes our estimate. There is some data that can measure this, and one data set shown here shows that there are about 540 proteins on average made per RNA in E. coli. So our estimate falls within this range. Now, the next example is to try to figure out how many ATP syntheses are needed to drive the synthesis, to produce the ATP to drive the synthesis of a cell. And we are very interested in knowing how, once we estimate that number, if they all fit into the membrane of a cell, because there is a limited amount of space in the membrane of a cell. And the ATP syntheses are large protein complexes. So we start out by collecting some information. So there are 230 revolutions per second in an ATP synthase. So an ATP synthase allows protons that have been translocated across a membrane to go back into the cytoplasm. And as the protons travel through the synthase, it has a rotor that rotates around at the speed given. For every revolution of the ATP synthase, you can make 3 ATPs. So that means that in an hour, which is roughly the doubling time of E. coli in minimal media, every synthase can make 2.5 million ATP molecules. Now, the computations or the estimates show here how many ATPs you need to make per gram dry weight. So that's 52 millimoles per gram dry weight. And we have an uptake rate of glucose under those conditions, aerobic growth conditions, of about 10 millimoles glucose per gram dry weight. The weight of a cell is given there. It's about 1 picogram wet weight, and about 30% of that is biomass. So we have about 0.28 times 10 to the 13 grams dry weight per cell. And we can now calculate that we need about 8.7 billion molecules of ATP per hour to drive growth. And growth here is a doubling approximately an hour. So now we can ratio the total need to the ATP production per synthase and estimate that we need about 3,500 ATP syntheses working at top speed to generate the 8.77 billion ATP molecules needed to drive growth. Now, the membrane carrying capacity of the inner membrane in E. coli is estimated to be about 200,000 protein molecules. There are about 2.5 million molecules, protein molecules in an E. coli cell, so about 10% are found in the membrane. The ATP synthase has 22 protein. It's a very large complex. So if I ratio 22 into 200,000, I can estimate that at packing, I have about 10,000, about 9,000 or roughly, give or take, complexes that can be in the membrane if all the protein carrying capacity of the membrane is used for ATP synthase, but I only need to accommodate 3,500. So about 40% of the protein carrying capacity of the membrane is estimated by these calculations to be occupied by ATP synthase. Bulk of the rest will have to go to cytochromes for the electron transport chain and for transporters of the substrate. Those three processes occupy most of the membrane space for protein during rapid aerobic growth. So these are two examples of how to carry out estimates based on the Fermi type estimation procedure, or just generally what engineers end up doing. So there is a database that helps you dig out numbers that you need for calculations available, called BioNumbers, hosted at Harvard with the link given at the bottom of this slide. And you can, for instance, get 920 different numbers out of it for E. coli and so forth. So this is a good resource to look for numbers if you need to do approximate calculations. So I want to continue now to look at real cells and make some observations. So first, the interior of a cell is very crowded. The dry weight of a cell is such that it is about 30% of the wet weight, so 70% of cells is water and 30% is biomass, and so it's extraordinarily packed. And here are some overall quantities that characterize E. coli and similar enterobacteria. The volume is on the order of cubic micron. The area is about 5 square cubic microns. That obviously should be to the second power, not the third power, so there's a mistake in this slide. The length of the stubby cylinder is about half to 0.8 microns in diameter and maybe 1 or 2 in length. The weight is a picogram, like I stated earlier, so 0.3 picograms biomass actually in there. 0.7 would be water. Size of the DNA, 4.5 million base pairs. I don't know why that says 10 to the third, should say 10 to the sixth. The number of individual protein molecules inside E. coli is about 2.5 million. That should also be 10 to the sixth, not 10 to the third. Doubling time about an hour. Mammalian cells, similar but bigger. So here we saw a slice through the key parts of a mammalian cell, going from the nucleus through the mitochondria and into the Golgi and the ER. And these are images from David Goodsell's book that is beautifully illustrated. A book of images from interior of different kind of cells. Here are some information about the overall composition of cells. Like I said, they are 70% water, 30% biomass. Of the biomass, you may have about half being protein, and so forth, as illustrated in this slide. Mammalian cells have a little more lipid content than bacterial cells because of the many internal membranes that they have. This slide shows classical data from Fred Neidhart at the University of Michigan, and he assembled this information here for the composition of E. coli that showed its composition as it was known 20 years ago or so. Now, of course, with omics data sets and other data sets, we are able to parse out the composition in even greater detail. To the lower right is a 4-panel figure of E. coli growing, or maybe not growing, but being on the tip of a needle. And I find this to be instructive because the resolution of our eye is roughly the tip of the needle. That's the first of the 4 panels. We can barely see that. And if you magnify that and look a little closer, we can start to see these bacterial cells growing on the tip of the needle, or just staying on the tip of this needle, I should say. And if you magnify it enough, you see these beautiful stubby cylinders that are the E. coli cells found on this needle. Timescales, pass on that. Here is a multi-scale diagram showing metabolism, protein synthesis, and cellular functions all in one. And this is one way of conceptualizing what that dense mixture of molecules inside the E. coli is actually doing. So that concludes some of the overall observations. Now we're going to proceed and try to estimate actual numbers for metabolism, and then for the macromolecular system. The first question we'd like to look at is what are typical metabolite concentrations in E. coli. Here's a histogram of the molecular weights of the known metabolites in E. coli, about 1,200 of them. The median is about 300 grams per mole. We can use that number and other assumptions to calculate the average concentration. If we estimate that there are about 1,000 different metabolites present in E. coli at a given point in time at this average molecular weight, we can estimate them. So out of the 1 gram per cc that E. coli is, 1% or so is metabolites. You divide that by 1,000, divide that by 312, and you come up with this estimate of the average concentration of 30 micromolar. And that's a low concentration, and we can do an interesting unit conversion here. We can ask ourselves how many molecules per cell or per cubic micron does 1 micromolar correspond to, and the answer is 600 as calculated. So the average metabolite concentration inside E. coli is maybe on the order of 20,000 molecules per cubic micron, which is roughly the volume of a cell. So that's a pretty low number. Now, when you go and measure, you'll find out that some metabolites are more abundant than others. The average concentrations for these are kind of in the range estimated, but some are very abundant, like glutamine, glutathione, ATP, and so forth. And since they are abundant, means that the others have to be even less abundant than the average. But our estimate is kind of in the zone. Here is a slide that gives some of the web resources or websites that have information about metabolism and metabolic quantities. Now we can estimate typical metabolic fluxes. We'll start by looking at the turnover times, and we had talked about in lecture 2 that you can estimate the turnover times by looking at the magnitude of a change, which we're going to take as being the concentration, and divide by the average flux or average rate of change to do that. So let's estimate some time constants. Let's first look at the relative rate of diffusion to reaction. The diffusional response times are estimated by calculating length squared over the diffusion coefficient, where the length is a characteristic length where diffusion is taking place, and that's a micron in this case. And if you carry out this estimate, you find out that you estimate the diffusion time for a metabolite in a bacterial cell to be, on the order of milliseconds. We can estimate maximum, and I should say that those rates are diffusion rates are very rapid and are too rapid to create significant spatial gradients inside the cell. We can estimate the maximum reaction rates achievable in metabolism by looking at the typical substrate and enzyme concentrations and using the maximum rates for the on rate constant. And that maximum rate is about 10 to the 8th per second per mole. And if you plug in the numbers, you find out that you would expect the highest fluxes in metabolism to be on the order of 1 million molecules converted per cubic micron. That's roughly a cell per second. And that is not a very big number. Turnover times, you can actually estimate them based on data. First, 3 billion glucose molecules are roughly required to build an E. coli cell. And if it's doubling every 60 minutes and the volume is 1 to 2 cubic microns, you can estimate the glycolytic flux to be on the order of 400 to 800,000 molecules per cubic micron per second, slightly below the estimate given on the previous slide. You can then estimate the turnover time of the glucose molecule by taking the flux and dividing it into the concentration that is measured. And if you do that, you estimate the half-life of glucose molecules inside the E. coli to be just a few seconds. Here are turnover times in red blood cell glycolysis. The flux through red cell glycolysis is sluggish, on the order of 1.25 millimoles per hour. And since it's a linear pathway, that is the flux that you use to divide all the concentrations in the pathway with. And that is what this table here does. And it shows that we have fast and slow turnover times that correspond to high and low concentrations in this case. So some metabolites have long half-lives, some have short half-lives, and this is the origin of timescale separation in dynamic models of metabolism. Now, I'm going to show you some measured response times that are of interest. Here is the energy charge that we've been talking about in this class and how it responds. And you see that the master timescale here is about a minute. A cell is disturbed in terms of its energy usage. The energy charge drops and then recovers. We will explore the nature of this response in Chapter 8 by building simple kinetic models of the energy for the energy charge. This is representative of a control problem that is called disturbance rejection. You perturb the system, and the system reacts to overcome that disturbance and return to its original homeostatic state. This is a key issue in control theory. The other problem we encounter in control theory or in general with the issue of regulation is the servo problem, and that is when you're trying to guide a system from one steady state to the next in a regulated fashion. Not to return to a steady state, but to move to an alternative steady state. Here are some measurements on discharge and recharge of the transmembrane potential in mitochondria, and you see that these dynamics are quite rapid. And in general, redox-related processes are fast and typically faster than what you see with the energy charge shown on the previous slide. Here is an interesting slide that shows the typical charge on energy transducing membranes. These charges tend to be on the order of 180 to 230 millivolts, and that is a high number relative to the physical limitation on bilipid layers. If you put a potential on them that is on the order of 280 millivolts, they simply become unstable. So cells are operating at what, 3/4 of that capacity or so. It is interesting to calculate a few things based on these numbers. If we take a gradient on the order of 230 millivolts, and we divide that by the thickness of a bilipid layer, lipid bilayer, which is about 7 nanometers in thickness, we see that that corresponds to about 300,000 volts per centimeter. Now that is a very steep gradient, and for comparison, about 1,000 volts in air will produce a spark. So this gradient is, 2 orders of magnitude greater than that. So the physics at the cellular level are quite different than those we are accustomed to at our size scale. Okay, estimating kinetic constants and their numerical values, that is hard to do, so you can go into collections and look at their distribution. And here is a distribution of Km values in Brenda, which is a database of kinetic values. And interestingly, the average Km value in this collection is almost the same as the average metabolite concentration we estimated, which is interesting. So on average, a metabolite concentration is close to its Km value, although that'll, of course, differ significantly from case to case. Here are the turnover times for enzymes, and here is 1 minute. So most enzymes are operating at a pretty high turnover rate as shown here and tend to have rapid time constants. I will not say anything about thermodynamics here. So we can summarize some of these quantities for metabolism. First, diffusion times are very rapid inside small cells, too rapid to be of concern. And this is the reason why we can apply the dynamic mass balances on a homogeneous medium. This is an assumption we discussed in chapter 1 or 2. The average concentration of a metabolite is about 30 micromolar. The maximum fluxes through metabolic pathways are on the order of 1 million molecules per cubic micron per second. Redox pools operate on fast timescales and respond quickly. Energy charge may be a little more slowly. The average Km value of an enzyme is close to the average concentration. Enzyme turnover times are fast, and typically metabolic transients are all happening within minutes. Power densities, we didn't discuss these, but the typical number for that is given there. We'll say a few things now about the rate of synthesis of macromolecules. And we're going to, of course, talk about DNA, RNA, and protein. First, DNA, talk a little bit about genomes. There's a genome map, circular bacterial genome, and here are some genome sizes based on sequenced information. Viruses are a few thousand to tens of thousands of base pairs in length. A few viruses are much bigger. Bacteria are on the order of a few million base pairs. Haemophilus influenza was the first organism to have its genome fully sequenced, 1.8 million base pairs. Thermatoga maritima is the one of the smallest free-living organisms with a genome size of 1.8 and so forth. The human genome, the haploid version, is about 3 billion base pairs in length. Here are some features of the E. coli genome, 4.5 million base pairs, 4,500 genes, as I stated earlier, about 1,000 to 1,100 base pairs per gene, and so forth. Some of these numbers are not that critical to us in this class. Typical protein concentrations, so let's talk about them. We can estimate them in a similar way as we did metabolite concentration. So cells are very dense solutions of protein. Like I said, 30% of the volume is biomass, and about half of that biomass is protein. And there is their typical density. An E. coli cell is maybe expressing about 1,000 protein at a given point in time. The average size of a protein is 35 kilodalton, and there's the number I just gave. 15% of the wet weight is protein, and so you can calculate based on this that the average protein concentration is about 4 micromolar, which corresponds to about 2,500 molecules of a particular protein per cubic micron, and that is a small number. And if there is 1,000 different protein present at 2,500 each in E. coli, you'll get a total number of protein molecules in E. coli of about 2.5 million protein. Remember, we estimated the ATPase earlier to be 3,500 copies per cell. There is the size distribution of genes and therefore protein molecules in E. coli, and that's a number I just showed you. And we can now actually, through proteomic measurements, determine the abundance of each one of those and calculate the average abundance. That is also shown here. As for metabolism, there is a number of web resources available for protein, and they are shown here. Okay, the next topic that that we need to be familiar with are the synthetic fluxes of macromolecules, and these can be estimated relatively easily. For instance, the E. coli genome can be replicated in 40 minutes with 2 replication forks. The genome opens at the origin, 2 DNA polymerases come on, and they zip around the circular genome and go to the terminus that is on the other side of the circle. And since we know the time, the number of replication forks, and the number of base pairs being made in those 40 minutes, we can calculate the speed of each replication fork, and it is on the order of 900 base pairs being made per fork. The rate of the RNA polymerase is much slower, and that number is given in this table of about 40 to 50 base pairs per second. And as mentioned earlier, the translation rate of ribosomes is about 12 to 20 peptide bonds per second into a growing peptide chain. Protein synthesis in E. coli, there is the rate of protein synthesis by each ribosome. Here is the number of ribosomes per cell, 7,000 to 70,000, heavily dependent on the growth rate. So based on those 2 quantities, you can calculate the total amount of peptide bonds produced in E. coli by multiplying just the number of peptide bonds per ribosome by the total number of ribosomes, and we can calculate there what is that, 80,000 to 1.5 million peptide bonds formed per cell per second, primarily a function of the number of ribosomes inside the cells. And this means that we can be making on the order of 1 to 3 million proteins per cell per hour, which is completely consistent with our estimate of about 2.5 million proteins per cell, and they can all be synthesized in about an hour to double the cell volume and have cell division. Finally, we're going to talk a little bit about cell growth and the phenotypic functions. So a lot of information is available about exchange rates, as mentioned in chapter 6. When you have an open system, the exchange rates are often amenable to measurement, and this here shows the nutrients flowing into E. coli, glucose in this case, and then the byproducts being made. The rates here are given in 2 different units, millimoles per gram dry weight per hour, which is what you would see in the fermentation literature, for instance, and that can be converted into simply molecules per cubic micron per second, and that's shown to the right in this table. Now, we can estimate a few things based on these numbers. 3 million protein per cell with an average size of 300 amino acids or so. Could be a little bigger, 350. Remember, when you do order of magnitude calculations, the numbers aren't precise. So here's the number of peptide bonds that can be made per second. And you can estimate the ribosomal density based on that being about 14,000. You can calculate also the total number of nucleotides you need to build a cell. You can estimate how many nucleotides you need to make the ribosomes, the tRNAs, and the messenger RNAs, and you can see those numbers here: 6 million for ribosomes, 11 million for tRNAs, and 2.2 million for the messenger RNA. The DNA, of course, we know the composition of it. It's about we need about 16 million nucleotides, and that leads us to an estimate of about 90 million nucleotides per cell that is needed. And similarly, we can estimate the number of ATP molecules required to drive synthesis, and the power density that corresponds to. This is a lot of detailed numbers that you don't need to follow, just the fact that we can calculate them, and we can then assemble such calculations on an overall spreadsheet as shown in here that allows us to estimate ATP production by ATP synthase, the uptake rate of glucose, the amino acids that are needed to be made for the protein, the bases needed for the nucleic acids, and so forth, and make sure that all of these components are made in a balanced fashion over the cellular doubling time. So we can basically rough out, by doing order of magnitude calculations, what the flux map of a cell should look like. And this is an example of such an estimation procedure. This slide here gives us an important number for and a number that those of you who go into industrial biotechnology need to be aware of. And that is the volumetric productivity of fermentations in fermenters. The numbers that one needs to achieve in terms of productivity to have an economical process is typically 3 to 5 grams of product like lactate per liter per hour. And that is a pretty high production rate. But if you can achieve that and you can drive that to high concentrations, and high concentrations would mean about 100 grams per liter, and if you can do that at a good conversion from the substrate, maybe get 50% of your substrate to be converted into a product, you will have a very nice economically feasible process in most cases. Just a couple of slides about metabolism in mammals and across the biosphere. This slide here shows two important things: organ size and what fraction of the body mass they are, and then their metabolic activity and what fraction of the oxygen they consume, or what fraction of body metabolism takes place in these different organs. So the heart, kidney, brain, lungs, the liver, and the metabolic organs all together are maybe 5 kilos out of a body weight of 65 kilos, so 150 pounds. What would that be? 130 pounds, about 143 pounds, I guess. 65 kilos would be 143 pounds. But if you think in kilos, about 5 kilos in a 65 kilogram person would be about 7.7% of the body mass. But these organs have very high metabolic rates, and they represent 72% or so of energy metabolism in the body. The volumetric rates at which some of these organs operate is not too dissimilar from that of microorganisms. The brain in particular has a very high energy transduction rate. So that's kind of striking, that only 7-8% of the body mass is responsible for over 70% of metabolism. So that's useful to know. Another thing that's interesting is that organism sizes range from a fraction of a picogram to a 100-ton whale. So this represents 21 orders of magnitude in size. And one of the more striking correlations in biology are shown on this slide over here, which shows the basal metabolic rate as a function of body size, forming essentially a straight line on a log-log plot, where the slope of this line is 3/4. So metabolic rate slows down with size to the 3/4 power. And this is a remarkable correlation. All right, so this has been a somewhat lengthy lecture of talking about numbers and where they come from up front. We talked a little bit about the order of magnitude estimation procedure, and we applied it to calculate a few things. I will state again that the ability to make order of magnitude calculations is very useful for a practicing engineer. So let's run through a few of these things. First, a bacterial cell has dimensions of microns and a weight of a picogram. So that's 10 to the minus 12 grams. That is just a tiny little speck. The interior of a cell is very crowded, very viscous, has a large number of molecular species in it. Cells are 70% water, 30% biomass, and most of that biomass is macromolecules. The metabolites are a small fraction thereof. Typical concentrations of metabolites and enzymes fall into the micromolar range to tens of micromolars, with a wide distribution around that mean. Some molecules being more abundant and a lot then being less abundant. Metabolites are present at about 19,000 molecules per cubic micron. Enzymes may be at 2,000 molecules per cubic micron. The diffusional response inside the cell is fast. It's 10 milliseconds or so, and is too quick to be of interest with respect to formation of gradients within the cell. So we normally treat the interior of cells or a cellular compartment as being uniform in a small molecule, in a low molecular weight molecule. Metabolic fluxes take place on the order of 10 to the fourth to 10 to the fifth molecules per cubic micron per second, so 10 to 100,000 molecules per cell per second. The top speed may be on the order of 1,000,000 or so. So that's a summary of numbers, some bio numbers for you that will be useful in various settings, and the procedure to estimate them is even more useful if you're able to carry it out efficiently. I have here at the end a few slides that illustrate a calculation that I think should be of great contemporary interest. And we're going to estimate the magnitude of the bailout package that the federal government extended a couple of years ago to our economy. And as you recall, it was on the order of $1 trillion, maybe $0.8 trillion. The deficit of the United States is now $1 trillion per year. And let's try to visualize that. Take a look at that. So we're going to start with looking at the $100 bill. There is a thin piece of paper that's worth $100. And if I stack them up into something that's about a half an inch thick bundle, we have $10,000. If I take 100 of those packets and stack them up, I have $1 million. There will be 100 packets of 10,000. Most of us will never own this much money, but that's the size of $1 million in $100. $100 million would fit on a pallet, and $1 billion would then be about 8 to 10 of these. And there we go. That's $1 billion. But $1 trillion is bigger than that. And we can visualize $1 trillion based on what we just calculated. And here is the image. This is $1 trillion. Look at the image of the person to the bottom left. And this double stacked set of pallets here is $1 trillion. So now you know what $1 trillion is the next time you hear that unit. And remember, the United States today owes 15 of these, $15 trillion. Professors shouldn't get political, but I think the young people taking this class should be very concerned about who is eventually going to pay this off. Thanks for your attention. This is the end of this lecture, and it is pretty dense in numbers and facts, a little maybe more similar to medical school than engineering, but many of the numbers covered are numbers that are useful and one needs to memorize. Thanks for your attention.